Very good morning. I am Dr. Vijayan, SRMIST, Trichrapalli. Today discuss about lactophenol cotton blue. Staining for fungus. Fungal stain an important laboratory diagnosis for identifying the infectious microorganism using lactophenol blue stain. Example, fungi equivalent mushroom, fungi eukaryotic organism that are classified into two broad groups. One is yeast, another one is mold. Their cell wall include chitin, the plant cell wall made up of cellulose, animal cell wall made up of peptidoglycan layers. They have a miniature structure. It is cannot seen without the own eye. By straining them with the appropriate dye, we can see through under a microscope. One of the stain like lactophenol cotton blue. The lactophenol cotton blue can be used to strain the fungal cell walls. It is a wet mounting media and the straining solution used to prepare the microscopic slides of fungus for the further studies. And fungal components are bright blue colors. Lactophenol cotton blue staining is a simple historical staining methods used for microscopic examination and identification of the fungus. The lactophenol cotton blue ingredients, the most popular techniques for coloring and examining the fungal is the lactophenol cotton blue wet mounting preparation already told, and it is simple to make. The following ingredients are includes. The principle of lactophenol cotton blue staining, the reagent lactophenol cotton blue staining, preparation of lactophenol cotton blue solution, procedure of lactophenol cotton blue staining, result and interpretations, limitation and applications will be discussed. This is the microplate for fungal growing media, another multifungus, the lactophenol cotton blue for fungal staining, and another lamp. Sir, I can. The principle of lactophenol cotton blue staining. The fungal spore cell wall is made up of chitin. The lactophenol cotton blue solution acts as a mounting solution and staining agent. The clear and blue in colors, the three main reagents. One is phenol. It is a disinfectant by killing and living organisms. Another one is lactic acid to preserve the fungal structure. Cleaning solutions that help to keep fungal structure intact. And aniline dye called cotton blue strains. The chitin is a cell wall of fungus, giving the fungus purpose for colors and highlighting and contrasting the structures. The finally, cotton blue to strain coat chitin on the cell wall and other fungal structures. The strain will given the blue color appearance, the fungal spore structure and such as hyphae. Another one material is glycerol, a streaky components that keeping the preparation slide specimen for dyeing out. Slides are prepared for microscopic fungus examination using lactophenol cotton blue solution as the mounting media, one of the staining agent. Fungal components are brightly colored blue, always wear glow and carry a biological safety hood when is performing these treatments. And the reagent lactophenol cotton blue staining prepare the 50 ml of lactophenol cotton blue staining solution is made up of. One is distilled water 50 ml, cotton blue, aniline blue 0.125 grams, phenol glycerol 50 grams, glycerol 100 ml, lactic acid 50 ml, 70% ethanol. The specific noted for lactophenol cotton blue solution is prepare at least 2 days before use. The preparation of lactophenol cotton blue solutions over 2 nights, leaving the reagent undisturbed to allow dissolving and maturation. The first day, dissolve the cotton blue is distilled water and leave to rest overnight. This eliminates insoluble dyeing. Second, using the productive glow and phenol crystal to lactic acid in the glass beaker and stirred using the magnetic stirrer until the crystals dissolve. And add glycerol, filter the cotton blue and distill water into phenol plus glycerol plus lactic acid solution and mix. Finally, the store at room temperature. The principles of lactophenol cotton blue. Cotton blue is used for medical mycology to examine the structure of fungal cell by making a wet mount. It is made up of two main components, methyl blue and lactophenols. The methyl blue is a historical stain that strained collagen blue in tissue slice, whereas lactophenol is a water-based mix of four components, including phenol, lactic acid, and glycerol, whereas the phenol is aided killing germs and lactic acid aid to the productive fungal structure. Glycerol inhibit the fungal cellulolytic actions. Where the preparation of microscopic slide, the methylene blue created vivid cerulean colors by straining the chitin in the fungal cell wall and lactophenol function in mounted. And preparation of lactophenol, the staining 2 days preparation. Example, cotton wool dissolve phenol crystals, glycerol additions and filtrations. Finally, the storage after preparation mix, keeping the room temperature and away from the light for subsequent usage. It is as 52 weeks shelf life from the date of production under these conditions. After purchasing, the lactophenol cotton blue should be stored at the original containers at 15 to 25 degree in cool, dark and dry place. Use these conditions, the product will be stable until the expiry date. They listed the product labels. Lactic acid, glycerol, phenol containing phenol crystals are dissolved, distill water, aqueous solution for 1%, analogous to cotton blue or alline blue. The procedure of lactophenol cotton blue staining. On a clean microscopic glass slide, add a drop of 70% ethanol. Add the fungal specimen to the drop of alcohol using a sterile mounting, such as incubation loop from the solid media, depending on the sample of use. Tease of fungal spores of the fungal sample and all used for needle mounters to ensure the sample is mixed well within the alcohol. Using a dropper or pipettes, add 1 or 2 drops of lactophenol cotton blue solution, prepare above. Before ethanol dried off, carefully cover the stain with a clear sterile cover slip without making air bubbles to the stain. Examine the stain microscopically at 40x to observe for fungal spore and other fungal structures. And procedure for lactophenol cotton blue staining: on the clear microscopic slide, drop 70% ethanol. A result and interpretation: the fungal spore hyphae and fruiting structure staining blue, white blue background staining pale blue. The nearby the slides will be appear for lactophenol cotton blue on the slides. For example, Aspergillus niger staining hyphae for fruiting structures, a delicate flue with a pale blue backgrounds. Trichophyton also staining for hyphae and the fruiting structure delicate blue without pale blue backgrounds. The limitation: it is only used for presumptive identification method for fungus. The followed other diagnosis tools such as biochemical and culture examinations. The components of solution should be used before expiry, including use of solution before it expired. The solution may be disturbed the original morphological functions. Stain can be only used for identification of fungus. The structure and note the young vegetative form of fungus. Stain can be stored for long time, the period of time. Quality controls: the lactophenol cotton blue solution should be ink blue colors, should be clear and free of insoluble particles. After staining the fungal cell with lactophenol cotton blue, fungal spores and hyphae view under microscopic high power 40x objective lens. The fungal spores hyphae will be pale to dark blue colors. Advantages: lactophenol cotton blue staining help in the identification of fungus sample as well as its structures. The applications: general information, the fungal is and structure will be identified. Identification is suspected fungal samples. Disadvantages: wet mount or lactophenol cotton blue strains. The slide cannot be preserved for long time durations. The strain does not allow to see the fungal cells early stage of development. It has the potential to affect fungal morphology. Lactophenol cotton blue is effective for recognizing and pre-summative identification fungus. Because a scratch stamp mount, it is just temporary. It will evenly disappear. Gonorrhea or spores may be expelled for gonadiogenesis, the cell and DS mounts. Precaution of use of lactophenol cotton blue, the only for in vitro diagnosis purpose. Overheating of organism during fixed process may be cause organism are similar to disintegrate. The timing of the decolorization procedure is determined by thickness of the smears, and enough allowance should be made for on the tube of specimen. Only proper trained and qualified lab professional will be use the products. Thank you.